Welcome to the next lecture on human genetics and biotechnology, focusing on human inheritance. I'm Wendy Nofke, and I will be giving your presentation today. Genetic traits are the characteristics inherited in the DNA. Some of these will have simple inheritance patterns, while others are more complicated. The Mendelian inheritance pattern in human is the simplest and easy to understand. These are traits that are controlled by a single gene, and they have two alleles, one is dominant to the other. With the autosomal traits, these are located on one of the 22 autosomes. A pedigree is a way of being able to show patterns of inheritance as it moves from generation to generation. To the right shows a simple pedigree where you have a male represented by the square and the female represented by the circle. An affected male for a particular condition will have the colored-in box. The affected female for the particular condition would have the colored-in box. And the line drawn through them indicates that the person is deceased. With the sex-linked traits, these are going to be traits controlled by a gene on the sex chromosome. Males are only going to inherit one X chromosome, and so they will receive a recessive trait that is always expressed on the male because there is only one allele. There is no other option. These are always inherited from the mother. They are also passed on to their daughters, but not their sons. This is because if the male passes on an X chromosome to the embryo, it will create a female. An example of this type of condition is color blindness. This is a slightly larger picture here of that same pedigree, where you can see this line drawn between the two individuals indicates a marriage or a union of the individuals. When the line comes down and then you have the additional lines coming down, these are going to indicate their offspring. So the siblings will actually have a line that goes up and then goes across to their siblings. And you can see this goes down where the siblings have gone on to mate with other individuals and then have created their own offspring. With non-Mendelian inheritance, the traits are controlled by multiple alleles or genes. You can have a multiple allele trait that will be controlled by a gene with more than two alleles. This provides a tremendously greater amount of genetic variation in the individuals. One example of a multiple allele type trait is the ABO blood typing. These are going to be the types of markers that are expressed on the red blood cells. With our red blood cells, we have type A, type B, type AB, and type O. What these indicate is the markers on the types of blood. So a type A person is going to have the type A marker. The type B blood is going to have the type B marker. The AB marker has both the A and the B marker. The O does not have any markers on their blood. The person will have antibodies in their plasma that are going to attack the marker that they don't have. So if a person has type A blood, they're going to have anti-B antibodies. The person who has type B blood will have anti-A antibodies. The person who has type AB blood will not make any, and the person who is type O blood will have both types of antibodies. So the antigens in the red blood cell will be the A antigen, because that is the marker on the cell for the type A blood, the B antigen in the type B blood type. The AB blood type has both antigens, and the type O has 0. These are inherited codominantly from both parents. So when we look at the blood genotype, what that means for the person who is blood type A is they either inherited A from both parents or A from one and O from the other. The person with type B inherited B from both parents or B from one and O from the other. Type AB inherited A from one parent and B from the other. And type O inherited O from both parents. With polygenic traits, these are controlled by more than one gene. Each gene has a minor additive effect on the allele. An example would be skin color, and because of this being a polygenic trait, that allows us to have the greater variation and different gradations of skin color. With pleiotropy, a single gene affects more than one trait. An example is a gene that codes for protein collagen, which is found in bones. This is also going to affect the ears and the eyes. This results in problems for this person with their bones, as well as having problems with the organs of the ears and the eyes. With epistasis, one gene affects the expression of another gene. An example is in the Labrador dogs. The black color is dominant to the brown gene for hair pigment. However, we do see yellow variations of these dogs. Yellow occurs when they have the gene that tells them not to deposit the pigment into the hair. If they have this second gene that says do not deposit pigment into the hair, it doesn't matter what color the other gene is, none of it will be deposited, so the dog will appear yellow. If they do have the gene that says to deposit the pigment into the hair, then the gene that is either brown or black will be expressed for the dog. There are several different genetic disorders. Some examples include Marfan syndrome, which is autosomal dominant, that affects both the heart and the bone. With sickle cell anemia, this is an autosomal recessive disorder that produces abnormal hemoglobin. With vitamin D resistant rickets, this is an X-linked dominant disorder that causes soft bones that are easily deformed. Hemophilia A is an X-linked recessive disorder that causes a reduced ability of blood to clot. When it comes to passing on genetic disorders, most of them that get passed on are recessive. The dominant alleles are expressed in every single individual. If it is serious enough, they may die young and not pass it on to their offspring. They will die in many cases before they have the chance to have offspring. With a recessive disorder, it's not expressed in those with only one allele. They end up being carriers that are more likely to pass the gene on to future generations. With the chromosomal disorders, these a lot of times will occur as the result of a non-disjunction or the failure of replicated chromosomes to separate during meiosis. One example is Down syndrome that affects the 21st chromosome. This is sometimes referred to as trisomy 21 because the individual ends up with an extra 21st chromosome. With Turner syndrome, you just get one X chromosome. With the triple X syndrome, the individual gets 3 X chromosomes. With Klinefelter syndrome, they get additional X chromosomes, but they also have a Y chromosome. So an example would be XXY or XXXY. The more additional chromosomes, usually the more severe the condition. Many chromosomal disorders are actually not viable. These are just a few that are. In order to diagnose genetic disorders, we have a couple of tools available. Genetic counseling can look at karyotypes of a fetus. It can also look at the history of the family with looking at which diseases were manifested. Amniocentesis is a way to actually test some of the extra fetals or extract some of the fetal cells and be able to test them. The disadvantage to amniocentesis is the fetus is already formed at that point. This will allow examination of chromosomes.